Welcome to this lecture on the principles of immunology. Today, we are going to explore one of the greatest medical inventions in human history, vaccines. Imagine your immune system as an elite army defending a fortress. If a highly dangerous enemy attacks out of nowhere, your soldiers might suffer heavy casualties because they do not know how to fight this new threat. But what if they could practice against a weakened enemy first? That is exactly what vaccines do. They safely train your body to recognize and destroy deadly pathogens without you ever having to experience the actual disease. Let us dive into how this brilliant biological strategy works. Now that we understand the basic premise, let us look at what a vaccine actually is in scientific terms. A vaccine is a biological preparation designed to provide active, acquired immunity to a specific infectious disease. It typically contains an agent that strongly resembles the disease-causing microorganism. This agent could be a weakened version of the microbe, a completely killed form, its toxins, or even just one of its surface proteins. When this harmless agent is introduced into your body, it acts as a training dummy. It stimulates your immune system to recognize the agent as a threat and destroy it, all without causing the actual illness. This practice run is crucial for preparing your body's defenses for the real world. Building on that initial immune response, the most important outcome of vaccination is the creation of immunological memory. Once your immune system destroys the vaccine agent, it keeps a permanent record of what that enemy looks like. If you ever encounter the actual virulent pathogen in the future, your body mounts a massive and rapid response, neutralizing the threat before you even feel sick. But vaccines do not just protect the individual who receives them. When a large portion of a community is vaccinated, they form a protective shield around everyone else. This is known as herd immunity. It is absolutely vital because it protects those who cannot be vaccinated themselves, such as newborn babies, the elderly, or people with severe illnesses. This concept of community protection leads us to an important mathematical question. Exactly how many people need to be vaccinated to stop a disease from spreading? To answer this, epidemiologists use the herd immunity threshold formula. Conceptually, this formula calculates the minimum proportion of a population that must be immune to halt an outbreak. In the equation, HIT stands for the herd immunity threshold. The variable R0 represents the basic reproduction number. This is the average number of people that one single infected person will pass the disease to in a completely susceptible population. If a disease is highly contagious, R0 increases. When R0 increases, the fraction 1 over R0 becomes smaller, which makes the required threshold much larger. Conversely, a disease with a low R0 requires a much smaller percentage of the population to be vaccinated to achieve community protection. Now that we have our formula, let us see this with a concrete example using one of the most contagious viruses known to humanity, measles. We are given that the basic reproduction number for measles is 15. This means one sick person will infect 15 others. We need to find the minimum percentage of the population that must be vaccinated to stop the spread. We start by substituting 15 into the denominator of our formula. The fraction 1 over 15 represents the proportion of the population that can safely remain susceptible. Calculating this fraction gives us approximately 0.0667. Subtracting this from 1 yields 0.9333. Finally, we multiply by 100 to get our percentage. Physically, this means at least 93.33% of the population must be vaccinated to protect the vulnerable. If coverage drops below this, measles will spread rapidly. Having seen how vaccines protect populations, we must now ask how we know a specific vaccine actually works in the first place. This brings us to another crucial mathematical concept in immunology: vaccine efficacy. Before any vaccine is approved by medical agencies like the World Health Organization, it must undergo rigorous controlled clinical trials. Vaccine efficacy measures exactly how well the vaccine prevents disease during these trials. It does this by comparing the attack rate of the disease in a group of unvaccinated people to the attack rate in a group of vaccinated people. This comparison is the absolute gold standard for proving effectiveness. Without demonstrating high efficacy, a vaccine cannot be manufactured or distributed to the public. To quantify this effectiveness we just discussed, we use the vaccine efficacy formula. Conceptually, this equation calculates the relative reduction in the risk of getting sick. Let us break down the symbols. VE stands for vaccine efficacy, which is always expressed as a percentage. ARU represents the attack rate in the unvaccinated control group, meaning the proportion of unvaccinated people who got sick. ARV represents the attack rate in the vaccinated group. If a vaccine is highly effective, the attack rate in the vaccinated group drops close to zero. This makes the numerator almost equal to the unvaccinated attack rate, pushing the total efficacy close to 100%. On the other hand, if the vaccine does not work at all, the two attack rates will be equal, making the numerator zero, which means the efficacy is 0%. Let us apply this formula to a clinical trial for a new viral vaccine. We are given that in the unvaccinated placebo group, 80 out of 1,000 people contract the virus. In the vaccinated group, only 4 out of 1,000 get sick. We need to calculate the vaccine efficacy. First, we find the attack rates by dividing the sick individuals by the total in each group. The unvaccinated attack rate is 0.08, and the vaccinated attack rate is 0.004. Next, we substitute these values into our formula. We subtract the vaccinated rate from the unvaccinated rate to find the absolute reduction in risk, which is 0.076. Dividing this by the original baseline risk gives 0.95. Multiplying by 100 gives our final answer. Physically, this means the vaccine reduces a person's risk of contracting the virus by 95%. Now that we know how to measure if a vaccine works, let us explore the different ways they are actually made, starting with live attenuated vaccines. In this category, scientists use the actual disease-causing virus or bacterium. However, it has been attenuated, which means it is severely weakened in a laboratory, so it cannot cause serious disease in healthy people. Because these vaccines are so incredibly close to a natural infection, they are highly effective. They often create a strong, lifelong immune response with just 1 or 2 doses. However, there is a catch. Because they contain a living pathogen, they can be dangerous for individuals with compromised immune systems, such as cancer patients. Common examples of this type include the measles, mumps, and rubella vaccine. If live vaccines pose a risk to some, how do we protect everyone else? This leads us to the second category: inactivated vaccines. For these vaccines, scientists take the disease-causing pathogen and completely kill it using heat, radiation, or chemicals like formaldehyde. Because the pathogen is entirely dead, it is physically impossible for it to mutate back into a dangerous form or cause the disease. This makes inactivated vaccines extremely safe, even for people with weakened immune systems. However, the trade-off for this high level of safety is that dead pathogens do not stimulate the immune system as strongly as live ones do. Therefore, these vaccines usually require multiple booster shots over a person's lifetime to keep the immune memory strong. The annual flu shot is a classic example. Moving beyond whole pathogens, we have a third category that includes subunit, recombinant, polysaccharide, and conjugate vaccines. Instead of using the entire virus or bacterium, whether alive or dead, these vaccines use only specific, isolated pieces of the germ. Scientists extract just the parts that the immune system can easily recognize, such as the protein coat or complex sugars on the surface. By focusing only on these essential antigens, these vaccines deliver a very strong and highly targeted immune response. Furthermore, because the rest of the microbe is completely absent, they minimize side effects and are safe for almost everyone. Like inactivated vaccines, they often require booster doses. Excellent examples of this highly targeted approach are the hepatitis B and human papillomavirus vaccines. The fourth traditional category takes a slightly different approach: toxoid vaccines. Some bacterial diseases are actually not caused by the bacteria themselves multiplying in your tissues. Instead, the damage is caused by poisonous toxins that the bacteria secrete into your bloodstream. Toxoid vaccines are created by taking these harmful toxins and chemically neutralizing them so they become completely harmless. These neutralized toxins are called toxoids. When injected, your immune system learns how to fight off the natural toxin. This means that if you are ever infected by the bacteria in the future, your body will instantly neutralize the poisons before they can cause any damage. The most famous examples are the tetanus and diphtheria vaccines, which require booster shots every 10 years to maintain your protection. To bring all of this together, let us look at this diagram illustrating the mechanisms of the four main vaccine types. In the center, we see a Y-shaped antibody, representing the ultimate goal of our immune system. Pointing towards it from the top left is the live attenuated vaccine, shown as a whole but weakened virus. On the top right, the inactivated vaccine is depicted as a whole virus that has been killed, marked by the red cross. On the bottom left, the subunit vaccine shows only the isolated protein fragments, completely lacking the main viral body. Finally, on the bottom right, the toxoid vaccine displays small geometric shapes representing the neutralized toxins. Regardless of the specific structure shown here, every single one of these approaches successfully trains the immune system to produce those vital antibodies. We have covered a lot of ground today, so let us summarize the key takeaways. First, vaccines act as the ultimate training program, safely teaching your immune system to defeat deadly pathogens without risking actual disease. Second, we saw how mathematical principles like the herd immunity threshold and vaccine efficacy allow us to scale individual vaccinations up to protect entire global populations. Finally, we explored the four main types of vaccines: live attenuated, inactivated, subunit, and toxoid. While they use different methods, from weakened viruses to isolated proteins, they all share the exact same fundamental goal: generating immunological memory. By mastering these concepts, you now understand exactly how modern immunology actively prevents pandemics and saves millions of lives every single year. Great job completing this topic.